nephron. And we'll start with filtration. And I have a picture of the nephron. Remember, you got a million of them. And collectively, the kidney will receive 775 mils of plasma per minute at that rate. Now, of that 775, here's how it breaks down. 125 mils per minute is filtered. For the rest, 650 mils of plasma per minute would go into the efferent arteriole, to the paratubular capillaries and the vasa recta for reabsorption secretion function. But let's focus on that 125 mils per minute. That flow rate of filtration will produce 1 mil per minute of urine flow for excretion when you get to the end there. So what that tells you is if you're filtering 125 mils and only 1 mil is excreted, that's an over a 99% filtration being reabsorbed there. So the thing I want to focus on is that normal GFR of 125 mils per minute is directly proportional to the net filtration pressure we calculated last time. Changes in GFR, this is what you want to minimize. You don't want it to change. Changes in GFR normally result from changes in glomerular blood pressure, but you want to regulate it at 125 mils per minute. The reason is, if GFR is too high, you're filtering too much. The filtrate flow is too fast, and so there's not enough time for needed substances to be reabsorbed. Needed substances cannot be reabsorbed quickly enough, and they're lost for the urine. GFR is too high, and the opposite is true. If GFR is too low, everything is reabsorbed, including like wastes you don't want, which are normally excreted. So that's why you have to regulate it at around 125 mils per minute. Can't be too high, can't be too low. That regulation, one mechanism we teach is that it's autoregulating. The nephron, each nephron can self-regulate its own GFR. Okay, as we'll see, to keep a relatively stable 125 mils per minute. But there's another mechanism in addition to the autoregulatory one that's hormonal, and that's the RAS mechanism, which we've learned before. But I didn't mention this one before. So in controlling GFR, one figure you should study for number one is this figure. So let's first talk big picture, then look at the nephron here. GFR, autoregulation. If you were to plot the GFR in mils per minute, and you were to plot it, well, what you would see is that it would pretty much be able to maintain 125 mils per minute over a wide range of blood pressure, 80 to 200. You would think that if blood pressure on the x-axis in millimeters of mercury, as we always talk about, if it's 200, if blood pressure gets really high, you would expect GFR to kind of like go super high, but it doesn't. And if blood pressure drops way low to 80, you expect it to drop off a lot. And it really doesn't. It pretty much stays almost even keel through a very wide range of blood pressure. If you exceed 200, maybe then it'll start to rise. And if you drop below 80, maybe then it'll drop off. But it's able to kind of maintain 125 mils per minute no matter what the blood pressure is doing because of these mechanisms we're going to go through, like for example this one. Let's first study this flow-dependent tubuloglomerular feedback. I'll just call it tubuloglomerular feedback. There's feedback between the tubulo, and the glomerulus. They talk to each other, okay? And if you just go through the steps, you can kind of see how that is. So if GFR changes in any way, if it gets too high or too low, this mechanism will autocorrect. I'll just go through the steps. Let's say blood pressure is high. So that would make the GFR flow a little bit too high, get above 125 mils per minute. Step 1. Increase GFR. So what's the mechanism to lower it? Well, what you got to know is how the nephron works. Number 2, you increase flow through the tube. Increase flow in tubule. So if you're increasing the flow in the tube, what they're not telling you, which they should, is what I said just prior. If you increase the flow in the tube, things are flowing by the cells very fast. So there's not enough time to reabsorb things. And let's focus on sodium. Not enough time to reabsorb sodium. Ions. Therefore, there's going to be more sodium in the filtrate because you're not able to reabsorb it. Increase sodium in filtrate. So understand that first because of this increased flow. Now, number 3, you flow past macula densa. The macula densa are these specialized cells. They're in the DCT. And let me point to the figure where they are. It's by the vascular pole, or it's right here. Well, you can see the number 3 there. That's what it's talking about. Again, the thing they're not telling you is the macula densa is a salt receptor. We call it an osmoreceptor. DCT. So this macula densa is an osmoreceptor. It detects salt levels, osmolarity, osmosis. It's going to detect that increase in salt, and it's going to respond to it, okay? Detects increase in sodium. And it's going to be able to talk to the afferent arteriole. It's going to release a vasoconstrictive chemical to constrict the arteriole, the afferent one. So number 4. So what the slide says is number 4, paracrine from macula densa to afferent arteriole. Paracrine response. Like endocrine, paracrine is when cells communicate with each other. They're in very close proximity. There's a secretion from the macula densa to the afferent arteriole. There's a vasoconstrictive chemical. Well, it's ATP. So from macula densa to afferent arteriole. So I'll say, well, they list it as a separate step, number 5. Vasoconstrict afferent arteriole. It's ATP that caused the vasoconstriction. So that's why I kept on harping in lecture, the lupin family, it turns back on itself, and the DCT, it always ends up by the vascular pole. So you can have this communication. Let's remember, the macula densa is close to the end of the nephron. So if there's changes that are happening, it can be detected at the end, and you can affect the beginning. Because when you vasoconstrict, well, increase resistance, decrease hydrostatic pressure in the glomerulus, that's going to drop the GFR. And just imagine in your mind, you squeeze this tube right here. Squeeze there, you're going to drop the GFR. That's all that is. So I'm going to put increase capital R, decrease GFR, if that makes sense. Any questions on that thinking? When you vasoconstrict one way. Okay, let me see if you understand this in Anna. I'm going to ask you this question. This is not how it works, but what if you were to squeeze the other tube, the efferent one? How would that affect GFR, up or down? It'll go up because it's hard getting out, so it's like more through there. So if you squeeze there, the inlet, you're going to decrease that. And so that's how you can autocorrect. Now the opposite is true. Let's go through that. Like for example, if you were, let's say the imbalance is drop GFR, and then go through all the steps, what's going to happen? Well, you're going to, well, why don't you do it? Just like with you and your neighbor in the next 1 minute. Just go through all the steps and just write how it's different this time. You should end up with, if GFR is too low, you want to raise it. So fill in all the steps in between in your notes. I'll check back in a minute. All right, the key is right here. This, how did you change this? If before you had not enough time, how did you change the phraseology there? You had a lot of time. So what should have happened to the sodium levels in the filtrate? It should have dropped, and that should have been detected by the macula densa, and it should have released, well, not something that vasoconstricts, but vasodilates. And that should let more blood in, and that should increase the filtration pressure to increase the GFR. Okay.
Professor?
Yeah.
So, for the opposite direction, is that what dialysis is for? Like the shunt?
For the opposite direction? I didn't understand that.
So, like, for vasoconstriction, like dialysis is either to reverse if it's too constricted or too constricted, right?
Mm-hmm.
So,
My understanding of dialysis is you're taking the blood and filtering it for them.
oh, okay.
Because there's some kidney failure where the kidneys aren't able to filter right there.
Oh, okay.
Okay. So I have to do some more research on the techniques of dialysis. That's the basic idea.
Yeah.
A machine is doing the filtering for them. Yeah. Okay. Well, let's also.
So I was just thinking of how like number 3 changed.
Okay. Number 3.
Yeah.
Right here. So I would just write, it detects a decrease in sodium. So if GFR is dropping, you decrease flow. If there's not enough time when it's too fast, there's too much time. So you have decreased sodium in filtrate because there's too much time. Things are flowing more slowly, so you just kind of reabsorb more, and you have a drop in sodium there. Is that clear enough?
Yeah.
Angelica? Okay. So that macula densa is part of something called the juxtaglomerular apparatus. And that's shown again here. So I have the picture unlabeled, but number 1 and number 2 is all I want you to focus on. Number 1 in this picture is the macula densa. On the previous slide, macula densa was a little purple patch. But here you see a little more detail. You can see it's part of the DCT. You can see it's densely packed columnar cells. Number 1, macula densa. So let's title this juxtaglomerular apparatus. So I'm only presenting 2 things, and sometimes books will present maybe a 3rd cell type, but I just keep it to these 2. The macula densa cells of the DCT, they're specialized cells of the DCT. They act as osmoreceptors, which is what I previously wrote. As we just got through learning, detects changes in sodium levels, right? That's an osmoreceptor, and sodium is the primary electrolyte you keep an eye on, and they're basically columnar cells of the DCT, and they're tightly packed. Normally, cells in the DCT are cuboidal, but they're so tightly packed and large. They're identified as macula densa. They're columnar cells. I'm going to go columnar ET cells because let's remember the tissue type, epithelial tissue. Now, number 2 is that's pointing to the specialized cells of the afferent arteriole called a juxtaglomerular cell. Sometimes I just go JG cells for short. They're also called granular cells, if you ever see that term in your book. Because the granules contain renin. These cells, they act as baroreceptors. They detect change in pressure. In particular, they detect a drop in BP because we're hardwired to increase BP if it drops. And well, the cells, they're actually specialized with muscle cells of the afferent arteriole. That's what number 2 is pointing to. Special, smooth muscle cells of afferent arteriole. And like, why are they special? What do they got? They contain, the granules contain renin. So that's the apparatus. We saw how the macula densa cells can alter the afferent arteriole. Let's look at this example here, this drop in BP thing. this flow chart, I know it's hard to see, I kind of outlined it on the side there. Let's say you have a drop in blood pressure. Okay, let's kind of go through this RAS mechanism again. This is like an extrinsic pathway of controlling GFR. Extrinsic mechanism controlling GFR, not 125. This is, well, RAS, I taught it a couple times before, renin, angiotensin, aldosterone system. So in this example, there's a drop in BP. That means you would have a drop in GFR. Okay, I already showed you previously how tubulo-glomerular feedback can raise it, but this is another way. So if BP is low, you want to raise it to boost GFR back up. So what this shows you is, I'm going to read from these tiny little boxes here. If you drop blood pressure, you drop GFR, and you have decreased sodium in the DCT. Drop GFR, therefore decrease sodium in the DCT. Okay, I'll let you think about that. Okay. Blood pressure drops, GFR drops. Okay, if GFR drops, what's happening to flow? Fast or slow? Slow. So you have more time to reabsorb. You remove more sodium, less sodium in the DCT. Right? And what we had said was macula densa, they'll pick up on that. And even this figure, they say macula densa osmoreceptors, the JG cells, they're more like mechanoreceptors. I call them baroreceptors. All right. Well, anyways, they have arrows that point to renin release. Renin release is what I'm writing here. But actually, which cells actually have them to release? The JG cells. Well, the figure they have both things pointing to it, but these are the cells that actually release the renin, okay? Anyway, when you get the renin released, that whole mechanism, renin will cleave angiotensinogen. I'll just call it angio. Tensinogen. Renin's a cleaver. And it becomes Ang1. And then, what I taught you before is ACE will cleave it to Ang2, that whole thing. And this figure reminds you, as I go through the outline here, that Ang2 has a couple of mechanisms. It can act through aldosterone, or it can act as a vasoconstrictor to increase blood pressure. Aldosterone is from the adrenal cortex. What you're supposed to remember is you increase sodium reabsorption, and water follows, and you increase blood volume. That's one way you can increase blood pressure, by increasing the blood volume. The other way is by vasoconstriction. I'll put VC arterioles. That increases the peripheral resistance. That's another way. You just squeeze the arterioles to keep more blood in the arteries. Both of those increase blood pressure, which was the end result. So you still need to know that physiology. Increase BP one way or the other. And so that's the RAS mechanism. When you increase the blood pressure, you'll correct the GFR. Okay. but you have to go through all this physiology to do it. So this is the last time you'll have to study it. No, it's the last test, so lecture exam 7. So here's a picture of those cells, and so like for example, let's see if you've been studying this. I have two things here. Is this afferent or efferent arterial? That's the afferent one. The reason why they cut that away, these are the JG cells. Those are glomerular capillaries. Now Bowman's capsule has two layers, a parietal and visceral. Which layer is that? It's visceral. See how they cut it away there? It's composed of podocytes. The parietal layer is the capsule part. There's the PCT there. If I kind of ask to identify anything here, don't put DCT. I'm going for Macula Densa, because that's where you find it in the vascular pole. They like to talk to each other, right? Here it is labeled, and they label it juxtaglomerular apparatus. But I told you these two cell types, Macula Densa, JG cells, or granular cells. Here's another picture. I like this one a little better because they do show you the podocytes, but they also show you those little filtration slits. It makes it a good filter. These are the JG cells if I point there. But if I point there, I'm going for afferent arterial as opposed to efferent. And if I point to here, I'm not going for DCT. I'm going for Macula Densa. Okay. Here's one more similar-ish slide. This one has the glomerular capillaries, and I guess that bluish color is the visceral layer of Bowman's capsule. But what if I said identify cell? Yeah, the podocyte, those little white things are like the foot processes coming off of it. You also have afferent, efferent arterial, but you see that red cell? That's a regular smooth muscle cell. But you see all those other cells that are polygonal shaped? Those are the JG cells. Those are special smooth muscle cells. Normal smooth muscle cells are like a spindle. And you can see for the efferent arterial, they have those spindle-shaped cells too. But that's afferent, that's efferent. You can see it's labeled over here. Okay. Now here, the model maker, I didn't make this slide. I would not call that DCT. What would I call it? Macula Densa. And so know that. Okay. And so that's the vascular pole. That's the tubular pole. And the first part of the tubular pole, well, it's the PCT. And that's what I'm going to talk about next. The PCT will receive all the filtrate. It's the first tube that will get to modify it. The PCT is at the beginning, the DCT is at the end. The PCT are your most active reabsorbers. So by the time you get to the end, there's not much left to reabsorb, and the cells reflect that. There's not as much cytoplasm. They don't have as much work to do. The PCT cells are your most active reabsorbers. This is an analogy. Reabsorption can be understood by simply taking a molecule like glucose, putting it on a ferry, and just going across out of the filtrate into the blood. That's reabsorption. Now, what I want you to know here is that glucose, it needs a carrier, right? Glucose, it's carrier. Now, the carriers are limited. So reabsorption and secretion, these processes, they have rate limits. Yeah.
Where are the PCT cells located?
Oh, just in the PCT, right? So,
What's the PCT? Oh, proximal complex.
let me show you a picture of home nephron. For example, here's the PCT. You see these cuboidal cells?
Yeah.
That's what I'm talking about. Okay. They line the tube. microscope, you have these big old, there's the tube, and the cells that surround it, they do all the reabsorption. And they have a lot of mitochondria, and they have a microvilli brush border because they're your most active reabsorbers. Oops. This falls off. So those are the features: more mitochondria, microvilli brush border. They have those things because they're the most active reabsorbers. Functionally, that's mostly what they do. And I'm going to focus on the reabsorption of sodium. Instead of having you memorize a long list of things that they reabsorb, let's focus on sodium in terms of the cell physiology. Oh, so again, the molecules in the PCT cells, reabsorption has rate limits. So reabsorption has rate limits. However, the function of the glomerulus is filtration. Filtration as a process, it has no limits. You can filter as much as you need to, as long as it can make it through the filter. So that's another thing you should know. Put in parentheses. Filtration at glomerulus has no limits. As long as it's small enough to make it through the filtration membrane at the glomerulus, it'll go right on through to the PCT. However, reabsorption has limits because it's dependent upon these carriers. There may not be enough of them, and the ones you have only work at a certain rate. And if you max them out, they call that TM, transport max, and they also call that saturation. You're saturating all the glucose carriers. They're all working, they're all used, they're all occupied. And you still have glucose. If you have too much sugar in your blood and it's all filtered, you may start to excrete glucose in your urine, which isn't normal. Okay, call it sweet urine. So that's the idea there. If blood sugar is at a reasonable level, euglycemic, you should have plenty of transporters. And you should have zero excretion in the urine. But if you have too much, you might max out and reach saturation, and you might have sweet urine. So the way cells work is not like this fairy analogy. This is really the cells you should be studying. Study a cell that lines the tube, and then study the molecules on each side of the membrane. Well, there's 4 sides, but study the side that faces the blood and faces the filtrate. Okay. There's the PCT cell. And studies, I'm going to leave a couple spaces for molecules I can draw. Anyways, this side faces the filtrate. They call that the luminal side because the filtrate is flowing through the lumen of the tube. Luminal membrane. And then on this side, you got the basolateral membrane, side opposite. So if you want to reabsorb a substance from the filtrate, which is coming through this side, passing by, you have to take it out of the filtrate and you have to bring it into the blood where the capillary is. Could be the vasa recta, could be the PC, let's say. I'll just say capillary on this side. So that's how they organize this figure. And to make it easy to understand, just go pass through a cell. These things are cell regulated. Stuff just doesn't leak through without cell work. So I'm going to focus on the reabsorption of sodium, which is one of the most highly filtered electrolytes. Put a number to it. 25,000 millimolar of sodium is filtered every day. So call that 100%. By the time you get to the end of the PCT, you're down to 33%. So you've already reabsorbed 2/3. And by the time you excrete, you barely have any sodium in your urine. You've reabsorbed something like 99.6%. Okay, so cells are very efficient at reabsorbing sodium. On this figure, I put some numbers in to help you understand the electrochemical gradient of sodium. So for example, in the filtrate, I go, let's focus on the concentration levels. In the filtrate, the sodium levels is 145 millimolar. However, in the cell, it's much lower, 15. Outside the cell, it's much higher, 145. So in terms of transport of sodium, think of how it wants to diffuse. If it wants to make it through the luminal membrane, high to low, that's favorable, right? It could easily do that if it had a transporter. Substances naturally want to go from high to low. What about from low to high? Nuh-uh. If it's going to pass through this membrane, you're going to have to like pump it against the gradient. Okay, and it's the same thing for the electrical gradient. On the filtrate, -3 millivolt. In the cell, -70. And outside the cell, pretty much 0 on that side. So, if you're a positive sodium ion, do you want to go where it's more negative? If you're positive? Yes. Positive is attracted to more negative. So, in both cases, to go through the luminal membrane, I call that downhill, right? Yeah. It's an expression. Just you're just following your electrical chemical gradient. Okay? But if you want to go across the other side, that you're going to need to do work. That's uphill. So let's start to talk about the molecules that will allow sodium to pass through both. All right. And so this figure, I emphasize that there's pumps that'll make the intracellular environment very negative. So I would just pay attention. There's this slide. I'm not going to go through the steps. I'm just going to focus on number 1, number 2. Okay, let's go do number 1 here first. Let's put a molecule in right here. Number 1. Okay, so first thing, that molecule is a sodium-potassium ATPase pump. It's in the basolateral membrane. What it's doing is it's a primary active transporter. It's utilizing the ATP. So what you see it doing, well, I'm using blue. They used red, but it's okay. It pumped out sodium in exchange for potassium. And then look what happens to the potassium. Potassium's exchanged into the cell, but then it leaks back out. I'll just draw an arrow with potassium going right back out. So the result of that hard working pump, it's utilizing ATP, right? Don't say you don't know that. I call it an ATP-based pump. It's got to be doing that, right? So the result is, if you're pumping out sodium, that's why that level is so low inside the cell. You're pumping it out. So the result is low sodium in cell. Also, in terms of electronegativity, you're pumping out a positive ion. And that positive potassium is also leaking back out. The end result is the inside of the cell is very negative. Sodium pumped out, positive potassium leaks out. The result is negative 70 millimole. It's very negative, and sodium, the sodium in cell, I should put low. Sorry, I couldn't put that. That's the result. So because sodium is low, and it's very negative, that makes the entrance through the luminal membrane downhill. But you got to put in a transporter because sodium is a positive, carries a full positive charge. It can't pass through the cell membrane by itself. So let's go, how about call this number 2. This is a 2. It's a sodium co-transporter, and it's in the luminal membrane. It's not a primary active transporter. It executes secondary active transport. Now the figure does show that. The solid red arrows are primary, and the dotted ones are the secondary active transport. This is a good example of a secondary active transporter is a good example of what we call the facilitated diffusion. The facilitated diffusion means it's diffusion, but you need this protein to help it along, to go through the downhill membrane. The work of the other molecule on the other side made it so entry through here is downhill. So sodium, because of this transporter, it just has a free ride. It's going downhill. So let me draw sodium entering the cell, being reabsorbed from the filtrate. It will end up in the blood. However, it's a co-transporter. You're taking along something else that's desirable along with it. The energy is provided by the sodium going downhill. So say, for example, glucose. Now glucose, it may be going uphill or downhill, but it doesn't matter. The energy is provided by the downhill sodium, okay? Something else desirable, you want glucose, and that will eventually be reabsorbed into the bloodstream. So this facilitated diffusion is a co-transport of sodium and glucose, or something else you want, right? Or some other nutrient. And I think that's the main thing to understand about how PCT cells work. The whole uphill, downhill thing, by creating this environment in the cell, it makes it very easy for things to be reabsorbed on the luminal membrane as things pass through the tube. Okay? So any questions on those 2 molecules? I want you to understand this in another way on glucose reabsorption. Let's say some data analysis. I want to get back to the idea of transporting something per unit time in mgs per minute. So you have a transport rate. However, what I previously noted for you guys is that reabsorption and secretion, they have transport rates, but they max out. Filtration has no max. I wrote that too. These processes depend on a finite number of carriers and pumps in the cell membranes. And when they reach their capacity, you've saturated and you've reached saturation or transport maximum for that particular substance. And that can be seen in graph form, say for example here. So let's just look at the graph. You have a concentration level in the blood of something, and it's going to be filtered at the glomerulus. So as it's filtered, it's going to be reabsorbed. And you can reabsorb when you're at a low enough level. But at some point, when the blood level gets too high, you're going to flatline. That's when saturation occurs. And even though you may have more being filtered, you can't reabsorb it all. So therefore, it's going to be lost in the urine. So again, the terms to keep track of are saturation and renal threshold. And write those down. My first idea, saturation. That's when you max out. Okay, all the carriers are used and they're all working at their max rate. Don't put TM. The M is max. You're maxed out. All carriers are used and working at max rates. So the units are some mgs, some milligrams of substance you're moving per unit time. Okay, but when you're saturated, that's the first point at which they're all used and maxed out. Now, renal threshold, that's a blood level. That's a plasma concentration. At which you zap all the carriers, you use them all. Okay, the first point at which they're all going to be used. Plasma concentration at which all carriers are first used up. So try to understand those two concepts by studying these three graphs. Okay. Well, I think it helps to kind of see this in picture form, these data. So we have our nephron processes, filtration, reabsorption, and excretion at the collecting duct. So here's our anatomy. To filter, substances must arrive through after an arterial, and it's going to be filtered at glomerulus, right? Then you have efferent arterial coming out. But we're not going to talk about that. We're just going to talk about things coming in. And let's say the substance we're going to filter is glucose. Let me draw, like, say, 3 little green balls. And let's say that that level is a low level that represents, say, I don't know, 100 mg per deciliter. And that enters the afferent arterial, and it's all filtered, and it's going to be caught by the Bowman's capsule and PCT, right? I'm going to draw the whole nephron as a single tube like that. let's pretend we're talking about the PCT. So when it's filtered and caught by the nephron, those molecules of glucose will pass by PCT cells in the tube, and they'll all be reabsorbed because you're well below threshold. So here's a capillary right next to it. So, I'll just draw all the glucose molecules, boom, being reabsorbed. And when you get to the end, you excrete nothing. I mean, you'll have urine, but just not any glucose, because it's all been reabsorbed, which is the normal function of the kidney. And if you keep increasing the level, at some point you're going to max out. Let's say you go 200. So I'm going to double the little green balls. They're all in the afferent arterial, and they're all filtered because filtration has no limit. If it's coming in, it's going through. It has no limit. And so when you pass by the PCT, you're still sub-threshold. They're all going to be reabsorbed. You're going to excrete 0. So when you get to 300, on this figure, that's kind of where things start to max out right there. But you're still functioning within normal parameters, but you're standing at the threshold, right? So let's go to 300. Let me draw 3 more green balls. They're all coming in the afferent arterial, and they're all going through. They're all filtered because filtration has no maximum. And when they pass by, when you're at threshold, you're still gonna get them all. You're still gonna have 0, but that's past that point. One molecule past that point, you'll start to excrete glucose. So if you just go to 400, just keep on adding to the picture there. Keep on adding more green balls. At this point, you've already maxed out. You cannot reabsorb that additional glucose. So finally, you start to excrete, have sugar in the urine, which can be detected in a simple test. All right, so why does this keep going up? It never levels off. What did I say about that? No limit. Just keep on going. So it all makes it through. PCT has limits. Normally, your blood should be around here. Okay, you should be reabsorbed 100%. But if you're hyperglycemic for whatever reason, I mean, it doesn't mean you're diabetic. I mean, it means you're diabetic if you fasted for 12 hours and you still have high glucose. You can just drink a big jug of Coke, and you may reach saturation because there's too much sugar floating in your bloodstream. All right. So, what I would say is, look at that number. That says 375, and it says TM. That's the transport rate that you can handle. Okay, but if you have to do more than that, you can't. That's the line that flat lines there. That's the limit. So that's why at 300, when you first reach saturation, that's the threshold. That's the 0 point at which you start to see excretion. So when you overlay all 3 graphs, you got this graph. That's why you just keep on filtering, and if you exceed your max limit, you'll start to excrete, and you kind of see that fall there. So when the blood plasma concentration of a substance, for example glucose, is so high, when it's filtered by the glomerulus, all of it can't be reabsorbed. So you got leftovers, right? And it's going to be excreted in the urine. And if it's glucose, you would have, it would taste sweet, but you wouldn't taste it, right? You just do a dipstick. I've heard the ancient Egyptians used to taste the urine. So we don't have to do that. Anyways, glucose is excreted at the high blood plasma concentration at which the filtered substance begins to spill is the threshold, renal threshold. So on our, okay, go ahead.
So if there's too much glucose in the urine and everything, wouldn't that lead to DKA, or is it just
DKA, what is
like
that?
diabetic ketoacidosis?
It could.
So then that can lead to renal failure, right? Because you're not filtering enough, or is it just fluid overload?
The main thing I want you to know is that the work of the cells, they can't keep up. Okay. Those other things, that they could be a cause. I have to think about that a little bit. In that multiple choice question, what was the correct answer in our example? A, B, or C?
C.
It was C. For those of you that do see it, you were looking there, right? But you don't see it. That's the threshold. That's the point at which you will excrete. Here's another question I typically ask for these kinds of questions, these kinds of data slides. What's the TM of those choices? Yeah, the 375, right? Choice D. Here's a question that's commonly missed. What do you think? And that's the common mistake, actually. Sorry, not to embarrass people, but yeah, okay, now you know it's not A. Erase, Scantron, what are you gonna put? Not C either. Okay, now you're down to B or D. It's not D. This is what you're using up. Those transporters are the limit. Okay. Yeah, that's a commonly missed question, and that's what you're using up. You're saturated because all the transporters are occupied and working at their max rate. Okay. So there you go. Now you won't miss it. All right. The next topic is acid-base balance. Actually, I changed my mind. This is a good spot for a break. Normally, I don't lecture after the break, but today I am. So come back at 12:30. I do want to continue on with acid-base balance and the